Dearly beloved, we are gathered here today.
Whoa. That is the wrong script. We're doing our genetics video today. Hi, I'm Kelsey.
Hi, I'm Jessie.
I'm Cameron.
And I'm Kara, and today we will be talking about the cure for the porcine reproductive and respiratory syndrome, also known as PRRS.
We are very excited about the opening of the National Bio and Agri-Defense Center and the research that will be starting. As our mission statement suggests, one of our main priorities will be working with the PRRS virus and eventually other industry-relevant issues, such as African swine fever. The PRRS virus is a small, enveloped RNA virus that contains a single-stranded, positive-sense RNA genome. A strain of PRRS virus mutates during circulation within a herd, and this strain spreads, causing it to become the predominant strain within the herd. Our research has used the CRISPR-Cas9 gene editing technology to cut out the problematic DNA at the indicated enzymes and replace it with the PRRS-resistant strand.
In working with the new Biodefense Center, we will aggressively study the PRRS virus and the CD163 gene that it affects. Let's take a closer look at the CRISPR-Cas9 system and how it works to protect cells. Normally, the PRRS virus is introduced into the cell where it would attach itself and attack the DNA within the cell, resulting in the common symptoms of respiratory and reproductive issues that its name implies.
The initial method of attempting to combat the PRRS virus was with a vaccine. But with so many different strains of the virus, it was virtually useless to vaccinate against one strain. Plus, with each vaccination, breeders still posed a threat to the virus. PRRS, in combination with other diseases such as mycoplasma pneumonia, can prove extremely detrimental to a herd. By taking PRRS out of the equation, we eliminate one of the main factors affecting swine herds today. This new edited technology not only prevents the issue of infection, but also can cover all the PRRS strains with one modification.
With a CRISPR-Cas9 modified cell, however, this is not the case. CRISPR, in tandem with the Cas9 enzyme, work together to provide immunity from a virus that is trying to infect the cell, in this case, the PRRS virus. The CRISPR DNA is interspersed with spacer DNA, that is a non-coding region of the DNA that aligns with the DNA sequence of the virus that is trying to infect the cell. The Cas9 enzyme is used to cut the DNA strand. The double-stranded breaks that occur as a result of this are then repaired, and this repair is created with the spacer DNA. The recombinant segment of spacer DNA is then immune to the virus which we are protecting against, in this case, PRRS. With this technology, we are able to combat the infection, equipping the cell with a strain of the virus in order to prevent the virus from infecting a cell whenever it enters. Is created with the spacer.
Plus, each vaccination still posed a threat to the with each vaccination, breeders still pose. I don't know why.
This repair is created with the inserted spacer DNA.